But I'll try to sort of pitch it in the language of turbulence. And what I'm going to discuss today in the next half an hour or so is, can low Reynolds number living flows be truly turbulent? So to give a sort of preamble, because this is a more inertial classical turbulence community, so essentially, active matter is abound. And for the purpose of this talk, at a very elementary level, I'm talking about systems which are out of equilibrium, systems which consume energy, in short, systems which move, systems which are living. And these sort of, about 25 years ago or so, and I'll go through that just a little bit, these sort of found a place in the community of statistical physicists because of very interesting properties that they show, and they show it at every scale. So here's a sort of pictorial representation where you see these systems at the scale of a meter going all the way down to microns. In this talk, I'll be talking about that last bit, when things are at the level of microns, and essentially much of this talk will feature about what happens to dense bacterial suspensions. And just again, for context or to compare numbers, I'll really be talking about the B. subtilis bacterium and the E. coli bacterium. You'll see why the E. coli is used here. So one of the very interesting things, and this sort of goes back to this paper by Wensing, actually, I mean, this was also seen a few years earlier, was the fact that when you have these dense bacterial suspensions in a two-dimensional configuration, they're all moving. They're moving very slowly, so the Reynolds number is close to being zero, essentially. But you do see patterns which many of us in this room have played with two-dimensional Navier-Stokes or two-dimensional soap film experiments. They seem to be very similar. So the question is, how do we sort of start modeling such two-dimensional suspensions, which have all these sort of very fine structures? So the microscopic approach, and as you'll see in this talk, this is just by way of preamble, especially for the students, but the microscopic sort of approach would be, essentially construct agent-based models which have interaction rules. So you, and sort of the real genesis of this kind of approach goes back to this paper by Witzek. So the other references are all here, where you kind of try and understand how these guys interact. So this is, for example, a movie from which Siddhartha is sitting right there, so made for me, which sort of shows that with very simple interaction rules, you can have agents which then tend to cluster, then tend to flock, and show very cooperative phenomena. You can also put them in turbulent flows, as my former student Mohit did, and you can see that you can again start to get, even in a non-trivial complex background, you can start to get cooperative behavior. But then within this sort of microscopic approach, you can then start to make effective equations even more complicated by putting in more realistic modeling, more realistic interactions. And essentially, you can start treating these little creatures as different sorts of rods or different shaped rods, which could be either polar, apolar, self-propelled, but all of these are essentially self-propelled. So that's where the activity comes in to mimic biology. And these sort of microscopic approaches have had a long history in the last 20 odd years. And this is a very nice review paper, which sort of lists out its many successes in trying to sort of understand the physics of living systems at several lengths. But in this talk, this is not the approach that I'm going to discuss. What I'm going to discuss is a more macroscopic approach. So here's a movie from experiments that some of us have been doing on an E. coli suspension. And when you see this movie, and if you see a soap film movie next to it, they look very different. They look very similar, right? So the question is, if I coarse grain my bacterial field, so we are sort of going to coarse grain our bacterial field. We lose information about the discrete agents, and now going to construct a hydrodynamic description of such a field. Here is probably an example of a coarse-grained. So when you take a macroscopic approach, and we'll come to those equations in a bit, then you see that you can start modeling these flows on the computer, and you get behavior which is very similar superficially, and we'll sort of see exactly in detail where this breaks down, to what one sees in the laboratory with real suspensions. So this analogy with sort of high Reynolds number turbulence, what I've been trying to motivate through pictures, sort of gave rise to this area of active turbulence, and this word sort of phrase came up in the work by Julia Yeoman and others in the last 7 or 8 years, or maybe 10 years. And what they sort of looked at is how formally is this bacterial field. This was the experiments of Wensing et al. This was their simulations of a coarse-grained description. Formally, they resemble what you would see from a two-dimensional high Reynolds number, inertial, without living creatures. They all look the same, but do they look the same at a superficial level, or is there some truth to this? And should high Reynolds number be considered sacrosanct as far as turbulence is concerned? So the two questions that I'm going to ask, the second question marginally in this meeting. The first question is, in what sense are these low Reynolds number living fluids truly turbulent? And which is perhaps a more important question to the poor bacterium itself, is how does the emergent complex flow benefit the bacteria, which is at the heart of it? So this sort of question was addressed by some of us in the group. So Siddhartha, who's sitting there, he's a postdoc at ICTS. Rahul and Martin were my former students. They're now postdocs respectively at IISc Japan and University of Genoa. And essentially, I'm going to describe the work in these papers. So here's a sort of physics in focus review of the work, which is quite nicely done. All right, so what is the hydrodynamic approach? So I won't go into the derivations. I mean, there are a couple of places where you find this, but essentially you first make the approximation that my flow is incompressible in the dense limit. It's an approximation, all right, we live with that. And then you write down an equation, a coarse-grained equation, which is rather similar to what you have for the Navier-Stokes equation. Notice that you are breaking Galilean invariance because now you have a lambda term sitting there. You're breaking conservation of momentum. And the signs on the Laplacian has changed, so to produce instabilities. But the most important thing in our story is the role of this alpha term, which essentially mimics how much energy the bacterium at the scale of the bacterium is injecting in the system. All right, we can later, after the talk, we can discuss about how these things are derived. So essentially, just to sort of keep track of numbers, in what I'll describe, the theoretical work, we will play with alpha in this range. These are just numbers, -8 to -1. And they correspond to bacterial speeds, which are easily realizable for the two sort of bacteria that I talked about, which lie in the range of about, of the order of between 10 to 100 microns per second. All right, so now comes the question, in what sense is this flow truly turbulent? So, and a good reference point for us is, of course, to go back to Uriel's book. And then I sort of make a little table here. So the first thing is that inertial turbulence at least admits a range of scales in which the energy spectrum seems to have a universal scaling law. I don't need to define energy spectrum to this audience. I will in a bit when I go through the calculations, but there seems to be a universal scaling law. In the bacterial flow problem, it turns out, and here's a reference from Frisch's group, but there were others around the same time, which shows that depending on the level of a bacterium, so the activity of the bacterium, whether they swim fast or whether they swim slow, the energy spectrum has no universal scaling range. All right. The other point that I'll mention here is, again, related to turbulence. I'm sort of focusing on issues which are in the high Reynolds number world, is that in real turbulence, in classical inertial turbulence, there is intermittency. And we've heard a lot of intermittency in the last week, and a bit, essentially, large tails and velocity gradients. But as the early works of from Yeoman's group and others showed, both in their models and in their experiments, these flows admit no large tails. So there is no large deviations, everything is Gaussian. And so to this sense, these studies would suggest that turbulent suspensions are not really turbulent. They might look chaotic, they might show vortical structures at the level of the bacterium at those scales, but they're not truly turbulent if you subject them to these sort of strict tests. What we will now show that this conclusion is only partially correct, and that truly inertial turbulence-like features actually emerge beyond the critical activity. And so, the remaining sort of 20 minutes of my talk will be to convince you that there is a critical activity which is experimentally realizable, where you start to see emergence of universality. So let's begin. So this is our starting equation, which is coupled with incompressibility. So we define in the usual way, let's look at the question of scale invariance, all right? So we define in the usual way the energy spectrum, and the two excellent references are Lessier's book for this and Uriel's. And what you see is you can sort of connect the energy flux through an effective time scale, which, let me remind you, in inertial turbulence is just a k to the power 2/3 range. And then you can sort of bootstrap yourself by connecting both the flux pi of k, which is just the integral of the transfer function, the advective transfer function, with an effective time scale. So what we did in this work, which is on arXiv and should be out in Nature Physics soon, what we did was we used a quasi-normal approximation in the standard way that one does. Uriel is already unhappy. And then one is able to write down the effective equation for the energy spectrum. All right, so far so good. How does non-universality and universality come in this game? It comes in through this assumption, and I'll try to sort of prove why this is correct. So what we will assume is that the effective time scale is some power of the scale kzi. Again, people here are from the turbulence community, so think what you would do for inertial turbulence by taking psi to be 2/3. That's the normal nature of the beast, and then you can sort of connect it very nicely just by plugging in the factors with the flux. So as the earlier papers showed, and as I introduced, there is scale invariance, but a lack of universality when the bacterium are slow and they're lazy like me. So where does this lack of universality come in this story? So the observation and assumptions, and this is from a whole bunch of data that people looked at, and this paper is a wonderful repository for that. What they found was that when you have small or low activity, the large scales are non-energetic. Essentially, you're pumping in energy at large wave numbers. The lower wave numbers are not excited, so all the energy is sort of concentrated at these large wave numbers. As a result of which, to cut a long story short, when you actually measure this effective time constant in this problem, you basically get no scale dependence in the range where you're looking for scale invariance. And when you put all of that together, and this is best done on the blackboard later, when you put all of that together, you come up with an expression for the energy spectrum, which has an exponent, which is certainly non-universal because it starts to depend on alpha. So the exponent that I showed you, where you have various little bits, where depending on different levels of activity or bacteria, you get different exponents, is essentially hidden in this argument, which is true when your bacteria is sluggish. So where does the emergent universality arise when you go to high activity? So when you go to high activity, one of the, it's really a combination of assumptions and observations. What you start doing is the following. If you notice, this delta is sort of a linear function in alpha. So as you pump, as the bacteria start to get more energetic, the large scales in the problem also start to become more energetic. So this assumption of a scale independent effective time scale starts to break down. And what you see is the emergence of two different time scales in the problem. One of this is the classical sort of thing that you would do in turbulence, which is a local eddy turnover time for shorthand. And then there is another 1/k time scale, which arises just because there is an activity which is leading to a polar ordering. And so you get noise, if you like, at all scales. And when you put this in and you construct an effective time scale from this by just doing a geometric mean, for example, then you get a time scale at large activity, which goes as k minus 7/8. So our measurements seem to confirm that these assumptions of the ansatz, it's not too bad. And more importantly, you sort of see a weak k-dependent flux, which was again something kind of motivated by earlier authors, that this flux is not constant in measurements they did in their lab. And you essentially see a weak sort of pi k, which goes as k38.
So could I ask you a quick question? What happened to gamma 2 in this argument?
Gamma 2 washes away because it's a very small number. So within the quasi-normal approximation, we can throw away gamma 2. They will reappear in some way here if you do the full closure, the result that we have. But gamma 2 will slave with gamma 0.
It can lead to strongly peaked spectra and other parameters.
Sure, the spectrum will always be strongly peaked, but within the theoretical approximation, gamma 2 is dropped away because it's at least 3 orders of magnitude.
We can discuss that later, sure.
Okay. So what we end up, putting all of this together is that when alpha is large, and I'll tell you in a minute what is large alpha in terms of real numbers, what we end up showing is that you get actually a universal scaling, no longer an alpha dependent scaling, which is k minus 3/2. Not minus 5/3, but minus 3/2. And then, I haven't gone through this, the actual sort of, this is the sort of leading order algebraic behavior, but you can do exactly a Lassier-like closure analysis. In the 70s, many in the Nice group have been sort of champions of this approach. And then you can actually find the full energy spectrum with the relevant exponential fall off. All right. So what's the first sort of take-home message? That there is indeed a critical activity, which in theory, in our theory, turns out to be minus 10. I haven't actually shown you how that's calculated, but we can do it on the blackboard if you like. It's a slightly more elaborate calculation. And what we predict is that for bacterium, which is slower than the critical activity, you have an exponent of their spectrum, which is activity dependent. The minute you cross over to a higher velocity, bacterium which has stopped being lazy, or which has had more food than it needs to, then you end up getting a universal exponent minus 3/2. So this sort of picture captures this rather well. The theoretical critical crossover alpha that we obtain is minus 10. Embarrassingly, the numerics suggest that it's around minus 5. And whether there's a factor of 2 that we missed, I'm not sure. But, and so to this extent, I think one could say that bacterial flows beyond the critical activity, it's still 0 Reynolds number flows. They have features which are starting to mimic real high Reynolds number turbulence. What about intermittency? So again, this part I will sort of go over just to show you the results without worrying too much about how we get there. But again, the same conclusion holds when you talk about intermittency. When you have activities which are sluggish, which are lower than the critical activity, everything is non-intermittent. But when you push your flow beyond that critical activity, while still having sort of order 0 Reynolds number, you start to see fat tails, and you can sort of quantify this in a meaningful way by, for example, measuring the kurtosis of these gradients, and you see a sharp increase beyond the Gaussian value, both in, when you do it scale by scale for alpha and as a function of alpha, as you see in this plot. So things do depart from Gaussianity, again, at around -5. So 2 take-home messages so far. Beyond critical activity, intermittency emerges in this game as a spontaneously emergent phenomena, and universality emerges. But it's not all good in terms of this comparison. So for example, when one tries to look at the growth of perturbations, so essentially what one does is we construct a flow field, and then we take a realization of that and perturb it very slightly. I think in this meeting, there have been a couple of talks which have dealt with this. And what we would like to see is how these perturbations grow or how quickly they decorrelate, right? So, let me also point out Sugan's work on this from last year, which is in the context of Euler, which formed the basis of our understanding. So what we do is when we actually calculate this decorrelator, we see that, and just to cut a long story short, when we look at the Lyapunov exponent, which is where I'm trying to focus with my laser pointer, it starts to grow and then it saturates at the critical activity and doesn't change. So to that extent, activity is not mappable to the Reynolds number. Ordinarily, you would, in inertial turbulence, you would have the Lyapunov exponent growing with Reynolds with a power of half, etc. But here, certainly, there seems to be an emergence of maximally chaotic states the minute you get beyond the threshold that you need to become, turbulent. So in the last sort of 10 minutes, Rahul? Yeah. In the last 12:30 lunch, right? Yeah.
Is it nominal?
In the last 10 minutes, I will sort of try to tell you a little bit about what is the implication of these maximally chaotic states for the bacterium themselves, all right? So, you'll find the details in some of the papers. So what we do is we essentially feed these bacterial flow randomly with Lagrangian or tracer particles. And, again, I'd sort of refer to Shweta, Rahul, Anupam, etc.'s paper. They've also looked at other aspects of Kiran, evening, but okay, Kiran, and because I have another of Shweta. Anyway, so what we look at is how are the individual trajectories of these bacterium going to behave? Do they sense the fact that there is an emergent phenomena which allows them to mix more optimally? So here is a sort of a representative snapshot of different trajectories which are artificially all placed at the center for easy comparison. And what you see is that for low activity, for example, if you look at the red or yellow, they appear like the normal diffusion process, right? They're sort of meandering, knotty, etc. When you start increasing activity, you start to see two kinds of trajectories. So here's a snapshot, here's a representative result from alpha -6, which is, again, to remind you, beyond the critical activity. What you see is that a bunch of trajectories have these persistent behavior. So they are persistent and straight, and a fraction of them actually have diffusive behavior. And these, and this I'm not going to talk about, but I'm just flashing in case anyone would like to discuss this. So these sort of are related to structures which appear kind of out of the blue in the flow, which facilitate these sort of persistent motion of these trajectories. So essentially, what we know is if I'm the bacterium, depending on which part of the flow I am, here, here, or here, I end up having three largely different kinds of trajectories which sort of give rise to the overall Lagrangian dynamics of this guy. So the implications for this, of course, lies in the well-known sort of measurement in statistical physics in the issue of mean squared displacement. So again, you will see the movie running, which sort of illustrates what I'm saying. So when we measure the mean squared displacement of these trajectories, at short times, you of course get a ballistic behavior. That's not surprising. At long times, you end up getting a t to the power psi behavior, where, let me remind you, psi1 is the usual diffusive case, and then you can have subdiffusive or super diffusive cases. So we went ahead and measured this, and what we found was that when we have a rather low activity, you have just boringly ballistic followed by diffusion. And we weren't the first to do it. There was a group in IIT Madras who had also sort of looked at this, and they discussed the crossover from ballistic to normal diffusion. When we were slightly sort of worried about why this was happening, we went back to earlier experimental results, and there was sort of two categories of experiments. Some which said that bacteria show anomalous diffusion, some which said bacteria do not show anomalous diffusion. So wherein lies the truth? of course, for what we are doing with our own biases, the work by Uriel and his group seem to be more in line with the sort of model that we were playing with in terms of the speeds of their bacteria, in terms of their activity, in terms of their suspension. And this sort of, and in this paper, sort of Uriel and others suggested that there is indeed anomalous diffusion in bacterial suspensions, and these are not surprisingly mediated by Levy-Walks. So why has it remained undetected in simulations? So what we believe is that the use of the activity term with the real physical velocities, that mapping was something which wasn't taken very seriously. And when we went ahead and actually pushed our activity to what one sees in the lab in real living bacteria, lo and behold, so I'm pushing activity up here, lo and behold, you immediately see just beyond the critical activity once again, anomalous superdiffusive behavior in the bacteria consistent with what the experiments would suggest. And these are, of course, mediated via Levy-Walks, which is quite good for us because it's a self-consistency check. And so we sort of went ahead and calculated the usual waiting time distributions, and you have the mathematical result, which sort of bootstraps all these exponents together to sort of convince yourselves that what we are doing is not entirely nonsensical. Of course, that's at the level of mean squared displacement, but you can also attack this question more directly in terms of first passage statistics. And I won't go through the calculations, but you can sort of recast this problem as a first passage problem with absorbing boundary conditions. And then you can again show that as you go from -4, which is below the critical threshold, to -6, which is above the critical threshold, the scaling function picks up this r to the TR being the first passage time with r to the power 2 by xi to again convincingly suggest that beyond critical activity, not only do you have universality of the scaling exponent, an emergence of intermittency, but also an emergence which was rather striking about anomalous diffusion, which hopefully is why living creatures actually do what they do. So I will end here because we have to break for lunch and sort of conclude by saying that there is an emergent universality in the scale invariance, which is accompanied by the onset of intermittency and maximally chaotic states beyond this critical activity. And more interestingly, and this is something that I haven't touched upon at all, but I could do it for those with a bias for disordered or glassy systems, we do find very strong evidence of dynamical heterogeneity in our trajectories, where you sort of condition the statistics of the trajectory in the normal way you would do in disordered systems. Right now, where are we? Right now, we are sort of gone back to the experimental drawing board, which is, unfortunately, and sort of performing experiments for inspiration on how to understand the role of geometry, confinement. So we are able to create three-dimensional drops, two-dimensional layers with incline, without incline, etc. in my colleague Shashi Thuttapalli's lab over at TIFR. And then rather sort of, fortuitously, we stumbled upon the issue just to keep the bacteria alive, to feed it. We started to detect certain interesting things when you have polymer additives in this flow and how they modulate this flow. Whether we need to be a little more careful when we do the theory at this stage in terms of momentum conservation and coupling to the solvent field more effectively, we will have to wait and see. For the moment, we are looking forward to the experimental data converging and giving us a clear picture. Okay, with that, I thank you and happy to discuss.
For a very nice talk. We have time for questions. We'll begin with Dhrupaditya.
So comment and question and okay, lots of things. So very interesting. So when you do the closure, always when you do the closure, it's perfectly okay to do it in the case where you don't, but the other thing, the fact that the scaling exponents are non-universal doesn't necessarily mean that it is not turbulent-like system. In 2D turbulence with friction, the scaling exponents are non-universal. Or if you have dust and feedback from the dust, then the scaling exponents are again non-universal. So I don't think that's a good argument for saying it is turbulence or not.
Mhm.
Okay.
Agree to a limited extent.
The second thing is that
Not fully, but.
when you are doing the closure and when you don't have intermittency, I think that's nice, that's halal. But when it is in the other limit, then you are on slightly tricky ground. And the last question is, I didn't know that there is a first passage time result for Levy work. How do you prove that?
So what we did was ignore the Levy work. We start with the Fokker-Planck with absorbing boundary conditions.
That's easy.
Yeah, so that's the way.
That's easy. But Levy,
we did it as a trajectory check to compare the exponent, because with the Levy you have
go ahead 2 steps. One more. No, further. Yeah, that expression R to the 2 by xi.
So this you can get from the Fokker-Planck with absorbing boundary conditions that you can write.
But for normal diffusion.
Yeah.
So
you don't have to worry about what's the trajectories per se, but you do it through the diffusion coefficient, right? So when you write the Fokker-Planck, you are not really caring about the underlying trajectories, right?
okay, fine.
Maybe you can discuss this in the Blackboard. Yasha, I mean, so the Fokker-Planck does not need any of these constraints, if that's what you were.
Any other questions, comments? Okay,
There's
make
one at
it
the
focused. Huh?
Yeah.
No.
The question is, see, it is not just the activity which is important. See, if you consider the density of the species, which, excuse me, density which you are introducing. So because the density determines the space scale and the interactions, and that the interaction.
I agree. So this is really when we are taking the high density limit. So I agree with you.
Yeah, higher low density and
so this is high density.
High density. Okay, and you can also introduce see low density living matter, low density living matter, but at the same time scale as the inertial scale. Supposing you introduce low density living matter, low density at the time scales related to the inertial time scale, then we see that will produce other time scales, the nonlinear interaction in the same inertial range, and still the Kolmogorov law may be valid.
Probably, but I don't know this at all.
Yeah.
Yes, thank you. You had a question?
I
Yeah.
had a couple of questions. To begin with, what was the time scale involved with, say, the bacterial population growth with respect to your flow time scales that you were? Because that tells you a bit about whether they're dying, growing, or they're suffocating, or they're
Good, true. So of course, here the flow field that you see, that is really the bacterial flow field. In the simulations, we don't, and the theory, we don't have to worry about deaths as much. In the experiment, we do. So the experiments typically are of the order of minutes. And then you again take the things out, put it in a motility buffer, again, sort of revive them, feed them. And then you again wait for their exponential growth in the population till they get in the centrifuge. And when you get enough of them, you put them back in. And not only that, what happened in the experiments, and Siddhartha is the real person, is that you also have to worry about the wear and tear, just mechanical, in flagella sort of breaking. So evidently, every time I try to pipette, I maim the bacteria and make them capable of swimming or walking. But so the experiments are done in the time scale of a minute or two. But you can get enough sort of temporal statistics I mean, this anomalous MSD picture we have from the experiments now reasonably, not very well, but reasonably. Yeah.
Yeah, I have 2 more quick questions. One is, I didn't understand actually how you quantified the activity level in the experiments and compared it with the numerics. And a second question was just more explorative. If you, I mean, even in the bacteria, they have different kind of locomotive at the micro scale. they do exhibit different locomotive behavior. It could be flagellar or it could be several.
Contractile, extensile, etc.
So one, I mean, maybe later, or even amebiotic would be very much like body deformation.
Mhm.
kind of. So one aspect would be to see how different form of microscopic propulsion mechanisms are going to manifest at the large scale.
Okay, that's a good. So let me answer, both a little and the rest. So what one does is essentially, as Rahul was pointing out, there are these sort of coefficients gamma 0, gamma 2, etc. Right? So using these sort of coefficients in the transport problem, one can construct the typical polar speeds of the bacterium, the typical length scales in the problem. Then you can go back to the experiment. So again, these would be non-dimensional numbers and say, I'm looking at E. coli, which has a dimensional velocity of blah microns per second, right? So you make a lookup table and then you sort of torture that lookup table well enough to come up with not a very exact mapping. All right, so for example, if you go back to earlier papers where they would sort of, completely in all honesty talk about, that up to some factors, these seems to be right. So the only thing then that's left is this alpha term, which is the activity term. And this we sort of compare with the polar velocities of the bacterium. And the reason we confine ourselves here is because, in the sort of different bacterium and different level of their sugar intake, we sort of found that their velocity sort of has a spread of roughly this, which corresponds in these rescale units, which are constructed from the time scales, from the length scales of these different parameters, which we can do it on the blackboard together. You come up with a range of alpha in this. So that's how we fix the parameters and are able to talk to an experimentalist, saying that when I mention alpha -5 as a critical value, this is the sort of speed that I'm looking at. Your other question was about the nature of the bacterium themselves. So within the course grade description, we don't have a very good handle on how the individual discrete agents look. But what we do have is through this lambda term, which depending on its value, it sort of at least separates bacterium which are contractile or which are extensile. So swimming behavior, so as you can see, it breaks Galilean invariance, momentum conservation has gone out of the window. And that's because we've sort of integrated out the degrees of freedom of the solvent in a very sort of, clumsy way. But what we are left behind at the end of the day is this lambda, which kind of tells me, is my bacteria a pushing variety or a pulling variety? So it's this fragile, whether it's, I really don't want to enact the action, then the analogy with Monty Python would become exact, but it sort of gives me a sense of whether, what kind of bacteria it is. So I don't know if I answered your question.
If I continue on the comment, have a comment on the follow-up, the question which was asked. Actually, you're dealing with micro scales and you have to use fluid structure interaction. It is not just the velocity which is important, but it's the fluid structure interaction is very important, because these bacteria, and depending on the geometry of the bacteria, linear ones, they can form vortices inside that, and so it is a fluid structure interaction problem.
Yeah.
And
Sure.
so you have to consider these equations of elasticity also.
I agree.
I mean, that might be more relevant at the scale of Frisch, which is the sorts of experiments she does. But perhaps shall we continue this over lunch?
I mean, Frisch, when you consider, excuse me, when you consider the geostrophic flow field, then Frisch is important. But if you are considering a flow experimental work in a pond, I mean, small tank, then even small 80, 100 micron.
I agree with you, except that
100 micron bacteria of the type of.
I don't know how to deal with it, so I sort of integrated out that.
Perhaps let's discuss this over lunch, and let's thank Shamruddhi again for a wonderful talk. And we will reconvene after lunch